and welcome to Plant Breeding and Genetics video series. Today we shall discuss about some of the current affairs in genetics, plant breeding and some general topics. So we are going to discuss about Breeding Informatics Network, Krita 3, recent GI tags approved, Pomegranate Genome Sequencing, Borlaug Award or Global Award for Dhan Shakti and many more. First of all, Breeding Informatics Network. This is one of the initiative of CGIAR. As you know, CGIAR is Consultative Group on International Agriculture Research, which is having 15 research centers worldwide and they are doing prominent research work. The centers I am naming here: Africa Rice or Africa Rice Center, Center for International Forestry Research, shortly called as CIFOR, ICARDA, that is International Center for Agriculture Research in the Drylands or Dry Areas, ICRISAT, International Crop Research Institute for Semi-Arid Tropics, then International Food Policy Research Institute, that is IFPRI or IFRI shortly, International Institute of Tropical Agriculture IITA, International Livestock Research Institute ILRI, then CIMMYT, International Maize and Wheat Improvement Center, CIP, International Potato Center, then IRRI, that is International Rice Research Institute, IWMI or International Water Management Institute, CIAT, that is the Alliance of Biodiversity International and the International Center for Tropical Agriculture, World Agroforestry and World Fish. According to CGIAR website, there are only 14 centers now. So, BRIN is the Alliance of Biometricians, Bioinformaticians and Geneticists supporting breeding programs. As you know, one person can't be specialized or very well versed in all the subjects, so collaboration of work is very much important for any improvement or breeding programs. This group coordinates to set up the CGIAR wide standards in analytics and decision support for all crops and implements best practice analytic support for breeding program. So this BRIN determines best practices through small focused network groups in coordination with CGIAR Excellence in Breeding, shortly called as EIB module leadership. BRIN aligns their network with breeding programs and their continuous improvement goals. The network group also coordinates with enterprise breeding system, breeding analytics and breeding platforms. This is very important. There may be they may ask for writing short notes in any of the competitive exams like IARI entrance or even in ARS mains exam. So you should have some key points about any of the technology or newly arrived techniques. This partner with the EBS development team in validating the implementation in EBS and also share same standards with breed base and BMS etc. partners. This aims to accelerate the modernization of breeding programs to increase the rates of genetic gain and shorten the cycle of varietal development and product replacement. In this video, I am going to give two short assignments for you. You should write down the location of all the CGIAR research centers and the definition of genetic gain and the importance of it and shortening the breeding cycle. In short, I may say that this particular technology is mainly focusing on speed breeding. Now about speed breeding, I have a elaborated discussion. You may go through it. So why we should go for BRIN? Because some centers lack resources and need support. Additionally, even when resources exist, the connection between breeding and breeding, and the breeding programs can be strengthened. Better resource and connections can result in improved experimental design, data analytics, precision support, ultimately positively impacting genetic gain and the development of better adapted varieties. As you might have even experienced, many a times we do better and excellent research, but we don't know how to interpret it. So to interpret and keep it in proper mode, we need the support of biometricians, and it is very important for paper publications in good journals. So, as a result, in the lack of coordination and standardization of analytics across centers and breeding programs, making it challenging for the donors to monitor the progress against the breeding program assessment tool, that is shortly called BPAT, recommendations and returns on investments. So, the goals of this particular technology is to increase engagement among members of the network, forming a network of co working community to solve the common problems together, ensure best practice recommendations and standards are set for all major decision points in the breeding cycle for all crops, supported by the Crops to End Hunger initiative, implement best practice recommendations in ABS breeding analytics, implement best practice analytics in breeding pipeline decisions. So there are so many outputs and opportunities of this system, like network group plans to align the breeding programs, continuous improvement plans and individual member work plans, then best practice and processes recommended and reported in standard requirement templates and SOP documents. Means here some manual kind of or guideline kind of things will be given to all the CGIAR research centers, so that even they'll collaborate with the sub centers, and there will be a standard and uniform protocol for all the centers. These requirement templates and SOPs feed into EBS product backlog and prioritize for target release versions. These standard documents turned into model parameter files for consumption into EBS breeding analytics. Best practice applied in breeding decisions and adapted in breeding process, which BRIN member are integral to the training and adoption activities. Then connect with the domain experts and integrator across the centers, programs, disciplines, public and private partners. Even it goes for PPP, that is public private partnership. Then conducting the regular trainings, webinars, networking events, and providing the resources to all the centers. This was all about our first topic discussion. Now coming to Kritagya Hackathon 3. Hackathon is the big gathering, you may say. According to this Kritagya means, in this KRI stands for Krishi or agriculture, TA for technique or technology, Gya for Gyan. That means it is a collaboration of agriculture, technology and knowledge. Kritagya 3 is for what purpose? This is for speed breeding. As in the previous topic also have already told you, speed breeding is given so much importance these days, so you should be thorough with this topic. This Kritagya event will give opportunity to the students, faculties, entrepreneurs, innovators and others to showcase their innovative approaches and technology solutions to promote speed breeding for crop improvement, to ensure overall sustainability and reliance in crop production in India. So this was a government initiative, like under NAHEP. Now you should give me the full form of NAHEP. Along with Crop Science Division of ICR will also help in enhancing the learning capabilities, innovations and disruptive solutions, employability and entrepreneurial drive in the crop science sectors. So in gist, it focuses on biotic, abiotic, stress breeding as well as speed breeding for crop improvement across the zones, regions by collaborating above said all the people like students, faculties, entrepreneurs, innovators, private partners, etc. Next, we shall discuss about the recent two GI tags. Recently, Mithila Makhana got GI tag. You might have heard about it. Makhana is a famous uh, uh, famous one kind of food, especially in the northern part of India. It is kind of rare food in southern part, but it is very good in terms of nutrition. Central government has awarded GI tag to this. With this move, growers will get the maximum price for their premium produce. So Mithila is a region, as you know, in Bihar. Makhana, growing it in underwater, like in lakes and in water sources, harvesting them, bringing them fulana. Yes, these are very kind of task, but still farmers go for it, and it is a very profitable uh, offense. Next is white onion of Alibag, or that is from Maharashtra, got GI tag. The famed white onion of Alibag in Maharashtra, Rega district, was given this GI tag, bringing worldwide recognition to its unique sweet taste, no tears factor, as well as its medicinal properties. So, geographical indication tag कब दिया जाता है? When it is specific to specific location and having certain properties which are only specific to that location, तभी दिया जाता है, and grown for a longer period of time. The soil of अलीबाग तालुक हैज़ लो सल्फर कंटेंट. तो यहाँ पे ऊपर पैराग्राफ में दिया हुआ है नो टीयर. नो टीयर मीन्स लेस सल्फर कंटेंट. The NABL approved lab test reports mention low pungency, sweet taste, no tear factor, low pyruvic acid, high protein, fat and fiber, etc. तो दो GI टैग्स के बारे में डिस्कस किया. नेक्स्ट इज डॉक्टर महालिंगम गोविंदराज अवार्डेड ग्लोबल अवार्ड. ही इज अवार्डेड विथ नॉर्मल बोरलॉग अवार्ड. ये किस लिए दिया गया? बिकॉज he has worked with Dhanshakti Bajra, which is rich in zinc and iron content. That is a biofortified crop which may address the hidden hunger and protein malnutrition kind of problems. And this was released during 2014, the first biofortified kind of pearl millet in India. He is a senior scientist for crop development at Harvest Plus and Alliance of Biodiversity International and International Center for Tropical Agriculture, CIAT. This award is for his effort to develop the biofortified pearl millet. Now, 5 new high-yielding varieties of wheat. In any competitive exams, they will majorly ask about the recently developed varieties. So, ICR ने develop किया है the Indian Institute of Wheat and Barley Research. तो इन्होंने claim किया है कि these 5 varieties may yield up to 75 quintals per hectare, helping the farmers to reap 5 to 10 quintals more than the average produce of 20 to 22 quintals per acre. तो कौन से हैं वो 5 varieties? BW 370 for high yield potential, DBW 371 recommended for early sowing, DBW 372 for this has yield capacity of 75 quintals, DBW 316 identified based on its superior quality and has been recommended for northeastern plain zone, eastern Uttar Pradesh, Odisha, West Bengal, Bihar, Jharkhand. Then DBW 55 identified based on its yield advantage and resistance to black and brown rusts. एक छोटा सा टॉपिक है अपेडा एक्सपोर्टेड जोहा एंड आइजुंग राइस टू दुबई. तो यहाँ पे एग्रीकल्चर प्रोडक्ट मार्केट एक्सपोर्ट एंड डेवलपमेंट अथॉरिटी जो है इंडिया का, विच इज़ रिस्पॉन्सिबल फॉर एक्सपोर्ट ऑफ मैनी ऑफ द कमोडिटीज़ और फूड प्रोडक्ट्स फ्रॉम इंडिया टू अब्रॉड. तो इन्होंने रिसेंटली असम के जोहा एंड आइजुंग राइस दुबई को एक्सपोर्ट किया. सो व्हाट इज़ द स्पेशलिटी ऑफ जोहा एंड आइजुंग राइस मीन्स दे आर हैविंग देयर ओन फ्लेवर काइंड. मतलब बासमती काइंड है लेकिन डिफरेंट काइंड ऑफ स्मेल है. For that reason they have high demand in Dubai or mainly the Muslim countries. Next is PBW 826, a new heat tolerant wheat variety. इसको PAU पंजाब एग्रीकल्चर यूनिवर्सिटी ने डेवलप किया, विच हैज़ बेटर हीट टॉलरेंस एज compared to other existing varieties available in the market. Introduced after 4 years of clinical and field trials, this witnessed 31% and 17% more yield than HD 3086 and HD 2967 varieties of wheat. अब हम डिस्कस करते हैं डिफरेंट बायोफोर्टिफाइड वेराइटीज़ डेवलप्ड इन डिफरेंट क्रॉप्स. ओके, सो इम्पॉर्टेंट है, इसका स्क्रीनशॉट आप रख लीजिए. राइस में CR धान 310 which has protein richness. Then DRR than 45, zinc rich variety. In wheat, WB2 for zinc and iron richness, and HPBW1 for iron and zinc. Maize में पोसा विवेक QPM 9 for provitamin A lysine. Pusa HM4 improved for lysine and tryptophan content. Pusa HM8 for lysine and tryptophan again, and Pusa HM9 for the same content. Pearl millet में HHB 299 for iron and zinc rich hybrid, AHB 1200 iron rich hybrid. जैसे मैंने धनशक्ति के बारे में ऑलरेडी बताया था, 2014 में रिलीज़ हुआ था, एंड दिस बिकेम वेरी पॉपुलर इन द सदर्न स्टेट्स, स्पेशली इन तेलंगाना, कर्नाटका, आंध्र प्रदेश. इस बेल्ट में पर्ल मिलेट बहुत ही पॉपुलर हुआ था, धनशक्ति. इवन फार्मर्स अडोप्टेड इट टू ए ग्रेटर एक्सटेंट. लेंटिल में पूसा अगेटी मसूर विच इज़ आयरन रिच वैरायटी. मस्टर्ड में बात करेंगे तो पूसा मस्टर्ड 30 फॉर लो एरोसिक एसिड वैरायटी. पूसा 00 मस्टर्ड 31 फॉर लो एरोसिक एसिड एंड लो ग्लूकोसिनोलेट. कॉलीफ्लावर में पूसा बीटा केसरी 1, that is beta carotene rich variety. Then in sweet potato, Bhu Sona for beta carotene, Bhu Krishna for anthocyanin richness. Pomegranate में है सोलापुर लाल for iron, zinc and vitamin C richness. Next, RNA interference technology से we shall discuss some of the applications or some of the traits improved. Like I am not going to discuss one by one here, only some of the important or recent one I'll discuss here. As you know, RNA interference technique क्या होता है? Blocking gene function by inserting short sequence of RNA that match part of the target gene sequence, thus no protein production. RNAi technology purely blocks or reduces the protein production. So आपने फ्लावर्स और टमाटो के बारे में सुना होगा, that reduced its ripening property and all. So here first one is enhanced nutrient content में LYE in tomato for increased concentration of lycopene, that is carotenoid antioxidant. Then DET1 in tomato for higher flavonoid and beta carotene content. SBE2 in wheat, sweet potato and maize for increased amylose for glycemic management and digestive health. FAD2 in canola, peanut, cotton for increased oleic acid content. SAD1 in cotton for increased stearic acid content. Then ZLKR or SDH in maize for lysine fortified maize. Second factor है reduced alkaloid production. COR and CYP8 to E4 are the two target genes for reduced alkaloid production. COR poppy, opium poppy में for production of non-narcotic alkaloid instead of morphine. Then CYP8 to E4 in tobacco for reduced level of carcinogen narcotin in cured leaves, because tobacco is also used in some medicines, as it is having many of the medicinal properties. heavy metal accumulation next trait है, यहाँ जीन फोकस्ड है ACR2 in Arabidopsis for arsenic hyper accumulation for phytoremediation. Then next trait है reduced polyphenol production, यहाँ पे जीन टारगेट किया है S कार्डिनिन synthase gene in cotton for lower gossypol level in cotton seed for safe consumption. As you know, gossypol एक toxic compound होता है. If any cattle consumes the cotton cake, gossypol will cause even up to death effect in them. So reducing gossypol is very important in cotton. Ethylene sensitivity, यहाँ पे 2 genes है, LEETR4 and ACC oxidase gene, both in tomato only. LETTR4 is for early ripening and ACC oxidase gene for longer shelf life because of slow ripening. Reduced allergicity next factor, ये peanut and ryegrass mainly आपने सुना होगा, peanut is allergic to many of the people. Okay, so ARAH2 for allergen free peanuts and LOLP1 and 2 in ryegrass for hypoallergic ryegrass. लास्ट वन है अनियन में reduced production of lacrimatory factor synthase. This is to produce the tearless onion. As I have already discussed in the previous slide, that in Maharashtra white onion got a GI tag for its tearless property or specific like that. तो नेक्स्ट टॉपिक पे हम जाएंगे, पोमोग्रेनेट जीनोम सीक्वेंस्ड. लॉन्ग बैक इन 2017-14 में ही चाइनीज़ साइंटिस्ट ने पोमोग्रेनेट जीनोम सीक्वेंस किया है. बट एज पर अगर हम इंडिया के बारे में बात करेंगे तो भगवा एक वैरायटी है पोमोग्रेनेट में, इसका सीक्वेंसिंग किया गया है. इट इज़ हैविंग 346.08 MB size with 342 scaffolds and an average N50 of 16.12 MB and N90 of 1088.62 KB. This assembly covers more than 98% of the estimated pomegranate genome size. तो इसका genome size है 352.54 MB. तो ये कहाँ पे किया गया है, ये पोमोग्रेनेट रिसर्च इंस्टीट्यूट है शोलापुर में, वहाँ से डॉक्टर NV सिंह एंड कोवर्कर्स ने. द जीनोम सीक्वेंसिंग एक्सपेरिमेंट वाज एक्सिक्यूटेड एट न्यूक्लियम इन्फॉर्मेटिक्स, मतलब ये पब्लिक प्राइवेट कोऑर्डिनेशन और PPP से हुआ है. दिस इज अ हैदराबाद बेस्ड जीनोमिक लैब. नेक्स्ट इज हरियाणा ग्रांट NOC फॉर फील्ड ट्रायल्स ऑफ BT कॉटन. तो BT कॉटन ऑलमोस्ट ट्रायल्स इंडिया में बैन हो चुके थे बिकॉज देयर वाज सो मच कंटेमिनेशन inculcated by the private sector. So Mahiko to conduct field trials on BG2 RRF, a herbicide tolerant and insect resistant variety of BT cotton. India has allowed commercial use of BG1 and BG2 GM cotton in the country, while the approval for BG2 RRF has been pending at various stages. The field trials are likely for the northern zone. Available BG2 RRF can provide protection against devastating pests like American bollworm. As you know, BT cotton जब भी डेवलप किया है, they are majorly focusing on bollworm resistance. So nowadays जब herbicide tolerance को अप्रूवल भी नहीं दिया है, तभी many companies were illegally promoting this in the private market. तो गवर्नमेंट ने काफ़ी सारा बैन लगाया. सो फ्रेंड्स, पार्ट 1 में हमने ये सब डिस्कस किया. सो आई विल कम अप विथ मोर करंट अफेयर्स एज वेल एज द multiple choice question video series also. Thank you for watching. If you have any problem or any doubt, please put it in the comment section. Like, share and subscribe to my channel. Thank you.